So the way I've organized my talk today is through a series of questions that we're going to go through and hopefully try to provide some answers to as well. So in terms of what I'm going to share, first we'll talk about, well, why study oxygen delivery and muscle blood flow in the first place? And then we'll talk a little bit about how muscle blood flow is regulated. Obviously, it's a general audience here today, so we'll kind of bring everyone to the same page. Then think about, well, what challenges does our system face? And how do we respond to those challenges? And as my title indicates, we typically respond pretty well. And then further, just look at what we're doing now in the lab and moving forward in the future. So first, looking at why study oxygen delivery and muscle blood flow. Well, first of all, why oxygen? And from the simplest perspective, it's vital to human life. And unless you're in my class or in my lab and really need to understand what this metabolic magic is, we can talk about this from a really simple perspective here in terms of taking oxygen molecules and then lots of really complicated stuff happens. And in the end, we produce ATP, or energy. And that ATP is what we use to do everything in our bodies, from fuel muscle contractions to help move different particles across membranes. And so without sufficient oxygen, this energy production is inefficient. And most of us, hopefully, if you're like me, you really appreciate efficiency in systems. And so we need sufficient oxygen around to maintain efficiency of this system. So how do we get it there? What is oxygen delivery, or what regulates oxygen delivery? Well, oxygen delivery is determined by arterial oxygen content and blood flow. So the amount of oxygen that's carried in our arterial blood and the amount of blood that's getting to a specific area. So why specifically muscle blood flow? Well, I mentioned that oxygen delivery is blood flow times arterial oxygen content. And in humans, arterial oxygen content is pretty much acutely stable. So it's going to be determined by total hemoglobin and saturation level. So hemoglobin, the protein that actually carries those oxygen molecules, unless we've suffered from blood loss or we're an elite athlete that's maybe taken some performance-enhancing drugs, typically our hemoglobin's pretty stable over a given period of time. And similarly, the saturation, or how much oxygen is actually binding to those hemoglobin molecules, is also relatively stable, unless we've had a great change in our environmental conditions, so going up to altitude or breathing air with less oxygen in it. So we can sort of take that aspect of the equation out and really think about the fact that blood flow is going to be the primary driver of oxygen delivery. So why the skeletal muscle in particular? Well, the skeletal muscle is going to be the largest tissue mass in the body, and it can increase its metabolic rate 100-fold. Meaning that this skeletal muscle blood flow is largely going to impact total peripheral resistance, which in turn determines mean arterial pressure. And anyone who has gone to the doctor's office, gotten their blood pressure checked, or stood up too fast getting out of bed in the morning can appreciate just how important being able to regulate mean arterial blood pressure is to our everyday life. So why do we care in general? I ask my students to question this all the time in the lab and in class. What's the overall bigger picture? Well, more than a third of United States adults have cardiovascular disease, including heart disease, heart failure, stroke, and hypertension. Cardiovascular disease is the leading cause of death in the United States, accounting for nearly 1 in every 3 deaths. And financially, we spend about $300 billion per year in direct and indirect costs on cardiovascular disease. And these stats are all a couple years old, so these numbers have likely increased. So to try to understand and attempt to understand the cardiovascular system as a whole is really important for sort of our overall health and well-being of our country and really worldwide as well. All right, so how's muscle blood flow regulated? We've talked now about why it's important, but how's it changing? So first, again, we'll look at the determinants of muscle blood flow. So we've got some equations here, if there's any engineers or math people in the room. Arterial inflow is going to be the change in pressure times vascular conductance. So the change in pressure that we're talking about here is the difference in pressure across the vascular bed. So going from the arteries, taking blood away from the heart, to the veins, returning the blood to the heart, that difference across the vascular bed is the pressure gradient that we're interested in. Vascular conductance looks really complex. You've got a lot of variables in here. It's determined by radius, viscosity, and length. And for us physiologists that like to keep things simple, it's kind of nice because in the human body, viscosity is also relatively constant. That sort of sludginess of the blood doesn't really change. And we're talking about a closed system, so the length of the system doesn't change. So we can actually simplify this equation so that the primary difference that's going to affect vascular conductance is the radius. And the radius that we're talking about here is the size of these arterioles, or these resistance vessels. And in the case of perfusion pressure being unchanged, again, we keep things pretty simple, especially experimentally when we're testing these ideas. If perfusion pressure is relatively unchanged, vascular tone, which is another way of saying that caliber or size of the blood vessels, is going to be the primary determinant of blood flow. So when I'm talking about changes in blood flow, what we're really talking about here is in these resistance vessels, so these deep, very small vessels within our skeletal muscle, are they getting bigger or smaller? And how are that size or radius changing, which then is going to affect vascular conductance, which then is going to affect flow. So how does this change? Well, again, we're looking here at a cross-section of one of those arterioles I just mentioned. The vascular smooth muscle, these are actually muscle cells surrounding the blood vessel. The lumen where the blood would flow is in the center here. These are the smooth muscle cells that are literally going to contract or relax to change shape. We also have a single cell layer of endothelium on the inner surface, which, as I'll talk about in a couple slides, it's actually really important in regulating blood flow. And again, these are resistance vessels in the skeletal muscle. If it gets bigger, as I mentioned, if that radius increases, we call that vasodilation. It's due to a decrease in the calcium concentration of these cells, leading to less contraction or more relaxation, less resistance, and an increase in flow. On the flip side, we can also have vasoconstriction. So here the radius is smaller, an increase in calcium, an increase in contraction, increase in resistance, or less flow overall. So how do we regulate these changes? Again, we're looking at this same cross-sectional view. So given a certain stress, like exercise or systemic hypoxia, breathing low oxygen air, these can activate the autonomic nervous system. So the autonomic nervous system can then stimulate the adrenal medulla, an endocrine gland that sits on top of our kidneys, to release a substance called epinephrine. It can bind to receptors, typically causing vasodilation or relaxation. At the same time, autonomic nervous system activates sympathetic nerve terminals to release norepinephrine, a different substance that binds to different receptors, actually causing vasoconstriction. So even right now, when we're just talking about the systemic regulation of muscle blood flow, we already have these competing forces. And what I'm going to try to hopefully get you to appreciate from this is just how complex this regulation is. In addition to these systemic or whole body responses, we also have a number of local factors. So again, that endothelial lining of the blood vessels is actually capable of producing various substances that are, for the most part, in a healthy person, going to cause vasodilation. And to even further complicate things, as we're learning more and more, we realize that the red blood cells, in addition to containing the hemoglobin that carries oxygen, that these red blood cells are actually also capable of sensing changes in oxygen delivery and themselves releasing substances such as ATP that can bind and affect this. So this is a very complex regulation, and we're still kind of figuring out how all this works. But I just want you to appreciate sort of all of the different inputs that's going into what we're interested in sort of this net result of what's the level of blood flow and oxygen delivery. So how do we know this? It's great, I can show it to you, but again, I always encourage my students to ask, well, how do we know this? How do we figure this out? Well, many studies have used isolated muscle masses such as the forearm. And so this is a picture from my previous lab. We have a subject laying on a table with their forearm extended. We're able to measure blood flow through the brachial artery, which represents how those resistance vessels downstream are changing. Zooming in, this is a more invasive way to address these issues, where we actually have a catheter in the brachial artery and in a vein, allowing for blood sampling or infusing different drugs or pharmacology, which I'll talk more about. We can cause different stresses, so we can have someone perform rhythmic hand grip, as well as, again, we can infuse different drugs. While this is an invasive setup, it doesn't need to be. So this is a picture from our lab now where it's a different way to measure forearm blood flow. We don't necessarily have to give drugs to tease apart and figure out some of these different pathways. So why use the forearm? Well, there's significant correlation between endothelial function or overall vascular function in the forearm and the coronary circulation. As you can imagine, it's pretty difficult to get at the coronary circulation unless someone's in the middle of a heart procedure, in which case they're not normally healthy, so we can't kind of establish baseline measures. Similarly, there's a relationship between the reactivity in the forearm and the cerebral circulation, another circulation that's difficult to test, but that we're obviously highly interested in. And again, we have these multiple non-invasive measures of forearm vessel function that are indicative of overall cardiovascular disease risk. So we can use the forearm as a model for other circulations and as a representative vascular bed to tell us more about the body in general. Some of the other reasons is that we can, if we want to, avoid systemic changes that can sort of complicate experimental findings. So something like hand grip exercise or just causing ischemia in the local area. If we were to do that to the whole body, we would have a lot of reflex responses that are good in terms of preserving sort of homeostasis and health in the body, but from an experimental point of view, make it really challenging to isolate specific pathways. So using the forearm allows us to do this. And again, in an invasive way, we can catheterize the artery to allow for pharmacodissection. And pharmacodissection is really just sort of a fancy word for saying to utilize different drugs in pharmacology to address specific signaling pathways and mechanisms to understand exactly how these responses are occurring. So the way that we can use that pharmacology to effectively figure out what's going on is that we have different drugs that we can give that essentially block the action of some of these substances that we think are playing a role. So a drug called phentolamine, we can inhibit what the nervous system is normally doing, and we can see how the person responds, and if that changes something, that suggests that that's playing a role and that's contributing to the response. So we have a couple of different pharmacology we can use to address those systemic or nervous system related factors, as well as a couple of different drugs. Here we're looking at a little different view. You've got the lumen with blood flowing, the endothelial cell layer, and then the smooth muscle cell layer below it. So again, that endothelial cell layer produces various substances that can aid in relaxation. And we have various pharmacology that we can inhibit the production of those things. And to date, it's been really interesting because what we've realized is that our body has a lot of ability to compensate. So a lot of times when we knock out a certain pathway, something else sort of revs up to take its place. And so it provides a really interesting challenge in trying to determine what exactly is contributing to these responses. One of the most challenging pathways in trying to understand vascular physiology has been that of hyperpolarization, or literally the electrical changes that are occurring in these cells. And so recently, we, in my previous lab, were able to translate and do some translational physiology from animal studies to human-based work to give a couple of different drugs to actually inhibit just specific ion channels and protein pumps that contribute to literally changing specific ions across the membrane. So this is where I get really excited about being a physiologist, because I get to think about things all the way from, feeling someone's pulse and heart rate, a really big picture idea, all the way down to literally just looking at the action of a single protein in a membrane of a channel pumping ions across and how that results in smooth muscle cell relaxation. So what I really like about this work is it really does span that spectrum of physiology from cell biology and electrophysiology all the way up to whole body systems. So just as an interim summary, what have we covered so far? Well, we know that oxygen delivery is vital to human health, and it's determined by muscle blood flow. We know that muscle blood flow is determined by the vascular tone of resistance vessels. And we know that the regulation of muscle blood flow is highly integrative and complex, and we can gain insight through studying isolated limbs. So moving right along, what are these challenges that my title, insinuated that we're able to match? So some of the challenges would be a challenge to oxygen demand. So we're looking at this balance of supply and demand. Well, something like exercise increases oxygen demand, again, because oxygen is needed to fuel metabolism. So we can study this by having someone perform a hand grip and lift a weight to increase metabolism. On the flip side, we can also create a challenge by reducing oxygen supply. In this case, doing so by reducing the arterial oxygen content through a subject breathing low oxygen air. So they're able to decrease the oxygen supply. So these mismatches are what the body is having to respond to. Also, we can affect this supply side by decreasing blood flow. So when you go and you have your blood pressure taken and they inflate that cuff up really high and your fingers get a little tingly because you can't feel them, no blood flow is going. We can do that experimentally, hold that occlusion for a certain period of time to allow for, again, a challenge to the system in decreasing oxygen supply that we need to increase blood flow to meet. These are all challenges that healthy people face. Some additional challenges, though, come with the inevitable process of aging, people who are sedentary or inactive, and certain disease states. So while I'm going to show primarily data from healthy humans that are able to meet these challenges, unfortunately what happens is that with age or certain disease states, a lot of times that ability to match is lost. And so that's where some of the more interventional-based studies that look to try to remedy this are focused. So again, how do we respond to these challenges? So I'll walk you through this graph here, and this is something that we'd all probably be able to predict. But we're looking at forearm blood flow both at rest and after 5 minutes of this rhythmic hand grip exercise. So if you look just at the black bar, the control condition, what we see is this significant increase. Again, we've increased oxygen demand, so to match, we must increase oxygen supply. And what these other bars represent are, again, using that pharmacology to try to understand exactly how that's actually happening. And so I mentioned those redundancies in the system. And what was cool about this data was that even though this is really only about a 30% change, prior to this, most of the time when people were exercising, the body found a way to still increase flow. So we think that these pathways, particularly these hyperpolarizing ones, are really important in regulating this ability to match. Moving on from exercise to hypoxia. So here we're looking at a normoxic, so this would just be normal air breathing, versus hypoxic, so low oxygen in the air. Again, focus first on the black bars, which is our young sort of control condition. And again, you can see that now we've lowered that oxygen supply, so in response, we must increase flow. And that's what the young people do. But also, as I mentioned, oftentimes with things like age, sometimes this ability to match is lost. And that's what we can see here in data from older individuals. So these are people age 60 and older. And when they're exposed to this hypoxic condition, what's occurring is that they're unable to increase blood flow. So that mismatch is still there, and that can have some detrimental effects, especially if you're talking in the long term or in populations like sleep apnea, where they experience these periods of hypoxia repeatedly throughout the night. When I was a master's student and wanted to impress my boss, I was like, well, what if we combine the two things? Let's do more. Let's do more. So what we've got here is from rest to normal exercise. Again, that same increase in flow. And then in another trial, we had them go from rest to hypoxia. So we've seen these data before in the previous two slides. So then my overeager project was, well, let's combine it. And what happens when we increase, and we now not only challenge with an increase in demand, but we also decrease the supply. And as we would have it, we're able to match. So you can see this is higher than the exercise. So again, our body is able to detect this additional need and adequately respond. Again, that wasn't new. I'm not that smart. Most of us weren't when we were master's students. So that had been shown before. Our new insight into this process was what are the mechanisms and how is it actually happening? And so again, we were looking at specific endothelial factors and saw that they did contribute to this compensatory rise. We're going to skip through this for the sake of time and move right along to ischemia. So again, here we're cutting off flow temporarily, then we're releasing it and seeing if the body responds. Focusing just the control condition and the closed circles, you can see this really impressive. So resting flow was about 3 or 4 mils per deciliter prior to occlusion. So when we release that cuff, we have this significant increase that then comes down and returns back to baseline over time. So we sometimes term this the reactive hyperemia. Hyperemia, the increase in blood flow in reaction to the period of occlusion. And what we were able to determine here was, again, looking at some of these specific signaling pathways, we were almost able to wipe out this response. So the applicability of this is that in populations that might have decrements in these signaling pathways, potentially they're exposed to that continuous mismatch more so than a healthy population. So what are we doing now? What are we doing in the future? Well, we're continuing to stress the system and determine responses. So this is my third year here, so we're getting the lab up and moving. And so one of the projects that we're working on is combining hypoxia and that ischemic stimulus I mentioned. And that's part of a Barry Summer Thesis Institute and thesis project through the honors program. We also have a student working on, again, looking at sort of some combined influences of that ischemic stress and a dietary stress. So when we consume sugar-sweetened beverages, and that can cause some detrimental effects on the system, how are we able to respond? Can we still match appropriately? And that's part of a graduate summer fellowship. So in the future, I'm looking to continue to build the laboratory capabilities in terms of measurements, as well as some of the devices that we use. So very willing to talk about collaborations for student projects and helping to design some of these more projects that allow us to cause more stimuli to the system, whether it be through suction or exercise, as well as move into some of these population-based studies that I mentioned in terms of looking at people who have impaired responses and or deficiencies in these pathways. And then ultimately looking at, once we understand them, what can we do to make those deficiencies better? So what have we learned so far? Well, hopefully our bodies, and specifically our blood vessels, have this remarkable ability to match oxygen delivery to demand. This matching is accomplished by the integrative regulation on both the systemic and local level. And human-based studies provide exciting learning opportunities for students interested in biomedicine. So it's really fun. I'm an alumni. My honors thesis advisor is here today, so it's fun to kind of close the circle and come back to this. And I think that as we're moving, and in our department, we're teaching a lot of pre-professional and preclinical students. And so supporting and encouraging this work that's really getting these students to be working with people hands-on is going to be really important for us.